Good morning. I'm Geulah Livshits, and it's my pleasure to introduce our next guests, Andy Holt, EVP Commercial Development, and Dr. Tim Kelly, President Manufacturing from AskBio, a wholly owned and independently operated subsidiary of Bayer. So the format for this session is a roughly 30 to 40-minute fireside chat. If you have any questions during the session, feel free to type them into the question box below the Zoom window, and we'll try to ask them on your behalf. So can you guys start by giving us an overview of AskBio and its manufacturing approach?
Great. Thanks, Geulah. So AskBio is a AAV-based gene therapy company that has been around for quite some time. I think people are probably familiar with our scientific founder, Jude Samolsky, who's one of the pioneers in AAV biology, who founded our company with Sheila McHale. And one of the things that Jude, I think, recognized in the very, very early days as he was cloning AAV for therapeutic use was the importance of manufacturing and the criticality of developing manufacturing technologies that would enable this technology to really have an impact on patients. So that's been a key focus for AskBio really since our inception is developing this manufacturing technology platform. And we made the decision a few years ago. One of the key things that Jude also recognized was a need to make this as accessible as possible, not only to support AskBio's therapeutic pipeline, but also to support AAV pipelines for other gene therapy companies. So that led to the decision to found Viral Gene Vector Core in 2017, a CDMO business which leverages AskBio's proprietary manufacturing technology platform and sort of makes that platform available to clients on sort of a license and royalty-free basis. So again, this has been sort of a key part of AskBio's DNA from the very beginning. And our clinical manufacturing facility that was certified in 2019 contains a capacity for over 50 batches per year presently at 250 to 500 liter scale. We're actually in the middle of an expansion to increase that capacity to over 70 batches per year at the same scales, 250 to 500 liters. And then that's a business that's supporting multiple client programs across different types of AAV applications, different serotypes, and so on. What we're really excited about now is our expansion with our commercial facility that Andy is going to lead us through a quick video tour of that expansion in progress. So we'll talk more about that facility as we kind of look through the pictures and so forth. But I wanted to provide that quick introduction to our CDMO business, Viral Gene Vector Core, and the AskBio AAV technology platform. So Andy, I'll let you crank up the video.
Sure. And just sanity checking that it's still showing, and the magic of the internet's working for everybody here. We'll get this going. You can see here, just to start, the reason to be jealous of where we get to work. This is San Sebastian, Spain, on the north coast, just on the border with France. It's a terrible place. Just before the session here started, we were talking about how the food and wine are all terrible and you'd hate to visit. Here, just to help orient a bit, the technology park, which is about 4 miles from the coast. So a easy run down the hill and a terrible run back up the hill, if that's your sort of thing. Here in the corner here, we have the Viral Gen 1 facility, which we gave a tour of when we were kindly invited to participate in this conference last year. And just out of screen here, past this castle, which it's a bank and a GMP production facility. I don't pretend to understand why, but it's a castle. Is also the doggy bone facility. This street here is called Paseo Micheletegui, and we'll be spending all of our time on the street here today because this is number 83, and our commercial facility is just up this hill that exits on the bottom of the screen at number 1, Paseo Micheletegui. So we're trying to build a little road here full of gene therapy. Here at the top of the hill, as I said, and throughout the video, you're going to see a mix of different things. Our facility opens at the end of June. Has been under construction since the beginning of last year, so coincides, unfortunately, well with the COVID epidemic. That being said, we're really proud of the progress that we've made. Wanted to show you that today. And you'll see a mix of videos, this sort of computer rendering of what it'll look like when it's fully complete, as well as a number of drone shots and time-lapse videos, as well as some walkthroughs of floor by floor of one of the modules. As we pan around here, you see the facility as complete is really a 3 facility. Each of these three modules, as they're called throughout, contains an identical floor plan. Each is 3 production suites that can maintain 2,000 liter production at a maximum single batch capacity. Integrated fill finish, warehousing, quality control, and office in each of these. So we're looking at a 320,000 square foot facility. Each of these, a total of 9 suites with 2K reactors in each. So as we go through, you'll kind of get some time in the office and the GMP and QC and warehousing, but just keep in mind it's a fairly modular and designed to be independent from one another to enable the most flexibility possible. You can see here as well, there's some below grade work, and to do that, here we start kind of the time lapse. You'll note the date in European format here as we're rocketing past the spring and summer. We had to dig a hole first here down below, and then we had to fill it up. Parking and warehousing are below grade, and here you see as module 1 and then 2 and then 3 start to rise out of the foundations that we built in the beginning of the year. You can also start to see sort of the floor by floor work here and the design of the facility. Quite a bit of work went into maintaining this construction. As you see, the dates continue to roll out here. We get into 2021 for the first time, and at this point, we have a real building. We have almost all of a real building. You'll see here the start of that roof line in module 1, and then this is a mezzanine for GMP access. This is the GMP floor, quality control, and then you'll see warehousing and quality control as we go down. So you'll see the structure echoed throughout as we kind of move through. And you'll start to see a better correlation at this point when you see this distinctive roof line on module 1, 2, and 3. This, for me at least, and this is from early March, is when it really starts to look like the GMP, you know, the building with the facade. I'll also briefly note that I should have let you know there's an amphitheater back here. This is the amphitheater, and so Virogen 1 is kind of right at the tip of this crane. The crane's pointing right at it down here in the scientific park. So you can see this is the road. This is Paseo Micheletigi that kind of goes down and loops around and comes back. So there's a path through the woods, but it's about 1 kilometer from door to door up by the road as we double back. So nicely situated, a smooth and easy transition from projects from clinical to commercial as we go through. Yeah, there's actually a better view of that amphitheater there. So down right there is where we're talking. As we move here, and you can see this is, again, from a couple of weeks later in March and into the building, we're gonna go top down, kind of floor by floor, and Tim's gonna take you through the GMP layout, which is probably the most interesting part, but we'll start here on the top floor, which is offices. We promised to put a client office right here in this corner to entice you to come look, because this is the only one that has a view of the ocean. We'll see who actually ends up with it, but fingers crossed it can be Tim or myself. But anyway, this is kind of that top floor underneath that sloping roof line and that nice glassed in. That's where all the office space is there on the top. Again, terrible place to have to work. And then we're going to go down into the GMP. We're going to skip the mezzanine because it's just mezzanine, and then QC, and then the warehousing as we kind of give you a tour here. This, and I'll try to get my cursor away so you can see all this. There we go. I'll turn it over to Tim here, if you don't mind, to help walk through kind of where we see the workflows and how GMP is going to look for us.
Yeah, great. Thanks, Andy. So first thing to note here, kind of, starting with sort of as the process flows through the facility, the main warehousing area will be, below grade here, as Andy mentioned. And I do want to emphasize again that these 3 modules are really 3 fully independently operating factories, right? So each will have its own dedicated warehouse down below grade, as well as this, kind of just-in-time warehouse, if you will, that's located within the actual clean room core. You can see the service elevator there in the top left to move materials from the actual main warehousing area below grade up to that sort of in process or just-in-time warehouse. We also note integrated cold room space and an autoclave, again, in each of these areas. So again, fully independent operating. The reason that we decided to do this with the 3 modules that were really fully independent was to just create as much flexibility to partner with clients as possible. So we can, basically sell slots the way that a conventional, CDMO within any of the modules. We can also consider more strategic arrangements where, we actually dedicate suites or dedicate entire modules. And again, if one were to do that, again, you'd have this fully independent, independent QC, independent warehousing, basically, a fully functional independent factory, if that were a potential avenue for collaboration. Each of these, you see the fill finish sort of area here, where we'd be actually filling the drug product into the validated container closure system. All part of the AskBio Pro-10 technology platform, including the formulation that's sort of optimized for stability of AAVs across serotypes. And that's another area, we'll talk probably a lot through the course of this around kind of the continued optimization of the platform. And so these are certainly areas that will continue to innovate and improve the platform, through formulation and other elements of the process. And so then moving into the actual processing areas, again, this probably looks familiar to most of you, fairly conventional sort of manufacturing design for commercial, completely unidirectional flows for personnel and materials and product throughout. A dedicated C-train room. All we do in that room is grow the Pro-10 cells. There's no product. So that is a space that is effectively shared for the 3 trains. We get the cells to then see the actual reactors, the production reactors housed in the upstream rooms. And then you'll note that the downstream rooms are quite large. These are admittedly sort of oversized, so that, we're sure that we can accommodate, sort of multiple generations of process technology, including, current conventional technology using ultracentrifugation for enrichment of full to empty capsid ratio. But we'll talk, I'm sure, through the course of this about our plans this year to implement chromatographic enrichment of full to empty and eliminate ultracentrifugation. But again, we really tried to future-proof the facility from a design point of view, so that even as we think about further advances in technology and things like stable cell lines expressing these vectors, that those processes will fit nicely into this facility, just as the current process with triple transfection of DNA works in this facility. So again, we really try to take the long view in terms of the design here, accommodate today's paradigm, but also be ready for the continued innovation that we're working on, that we know many of our colleagues in the industry are working on as well.
Thank you, Tim. To see what these look like right now, as of early March, here in Module 1 and 2, you can see, for those of you that are engineering nerds, nice high clearances here with everything up above, fed from the mezzanine that's directly above GMP. Really, the idea here is to be a very smooth transition from our clinical facility, which is a 3-suite facility with up to 500-liter capacity. It has a lot of the same integrated quality control. We want the methods to feel the same. We want the projects to feel the same, the product contacting surfaces to be the same, just to make life as easy as possible. And here you get a bit of a visibility on the below-grade warehousing area. As Tim alluded to, this is the each unit of the building has its own access. And you can see here physically, there's multiple doors in and there's a subdivision between each of the 3. But this is a full floor plate worth of warehouse, because I don't think any of us have ever been in a manufacturing facility that had too much storage. So we were trying our best to make sure that as Tim alluded to, that we really future-proof this as much as possible. Here, just to link it back briefly, this is a bit, this drone footage, you can actually see here the office space. This is the mezzanine GMP, and as we go down, in case that was of interest to you. But as we kind of round home here, what we have, or what we hope to have here as we open, is the most flexible GMP capacity for AAV manufacturing in the world. Virogen's really just AAV, and so we tried to leverage this sort of expertise and experience we have, but at the same time acknowledge that this area is really dynamic, and what we're doing now isn't probably what we're going to be doing in 5 years, certainly not in 10 years. So with that, I'll stop sharing and open any questions, and looking forward to a conversation here.
Fantastic. Thanks for that peek into the new facility and the future and what it's going to look like and the capabilities there. So clearly AskBio's facilities are advancing. You mentioned future-proofing the facility. So at a high level, how has AskBio evolved over the past years with respect to its capabilities and areas of focus within manufacturing? And kind of what have you learned from the process that you're taking forward in this next facility?
Yeah, thanks, Geulah. So, I mean, obviously, one of the key areas of evolution is the commercial facility. You know, this is kind of a key expansion for Virogen, so that we can provide that kind of complete life cycle partnership for our clients. We have many clients that we're supporting for phase 1, phase 2 manufacturing. The current facility is actually certified by EMA for phase 3 manufacturing as well. But we certainly expect many, if not most, of our clients to transition to the commercial facility for their phase 3 supply, and then of course for their PPQ and commercial supply. So that's been certainly a very, very strong focus for the business, is to ensure that we have that continuity of supply. We have that robust sort of partnership model to take these products all the way to commercialization, really start to impact patients. I think beyond that, from a technology point of view, certainly I kind of alluded to, it's sort of part of our DNA, kind of always attempting to innovate in this space. A big chunk of our organization is really focused on just continuing to optimize our technology platform. So that includes, I mentioned the shift from centrifugation to chromatographic enrichment of full to empty ratios, which we will implement at Virogen this year. As I mentioned, we're also have a lot of activity going on in, I guess, what I would characterize as process intensification, working to further enhance what are already sort of best-in-class, industry-leading productivity in terms of titer and product quality. But we're seeking to basically get a 5-fold enhancement on those best-in-class productivity sort of metrics with this additional process intensification, media optimization, cell line density optimization, things that are going to allow us to do even more as far as volumetric productivity of the process, particularly important as we transition into commercial production for these high dose systemic administration types of products. So those, I think, are probably some of the key areas. Also, probably talk at some point here about our focus on analytics. This is a really strong commitment that we have to really, getting to the point where we can really consider these to be well-characterized biologics. I think in the viral vector space today, I think we're a bit limited in our ability to really understand, these products analytically, and we're really trying to push the envelope there to have, our AAV vectors be as well characterized as other conventional biologics like antibodies and therapeutic proteins.
Got it. Great. So yes, maybe let's dive into the technology itself in a little bit more detail. You mentioned a number of areas where you guys have been advancing the platform. So maybe you can talk a little bit more about what you've learned in terms of the advances of the PRO-10 cell line and its improvement in AAV yield capabilities, and the other attributes of the manufacturing technologies that you've incorporated and how they improve yield and other parameters versus what else has been available.
Yeah, I think PRO-10 at the heart of it is a fairly straightforward thing, right? It's important, and I think a lot of the foresight that Josh Grieger and a lot of the team that really were integral in creating the platform had that was has made life really easy for us since is that it's a 293 clonal derivative, which is a thankless task to find a high yielding clone of any cell line. But we started with a really clean lineage. We've got that cell line early, and we've kept it very traceable. So from a regulatory perspective, there's really no issues bringing that into commercialization. We were very careful about adapting it to animal origin free and chemically defined environments, and getting it into suspension, of course, which lets it be scalable, but also really minimizes a lot of the input risk. I think I'm personally grateful to never have to care about the fetal bovine serum market ever again, because that's not one I'd prefer to participate in if I don't have to. And so a lot of that planning and foresight about how that was created has really enabled a lot of flexibility on the back end. And we made some strategic choices. I think the cell line itself obviously is really good at making AAV while being quite transparent about where it's come from and where it's going. But then it does enable kind of both in terms of the harvest and the purification, a really nice firm foundation to use industry standard techniques. I think, we're not particularly shy about, moving to chromatography as our only method, but we've been using ultracentrifugation for a number of years because of its ability to deliver really, really enriched populations of full capsids. I think the industry is really coming around to the idea, intuitive though it may be, that more is better in terms of full capsids. And as a group that's been consistently releasing, batches with at least 70% full capsids, if not quite a bit higher for a number of years, I think we're happy to see that quality attribute evolve. I think it raises some challenges as you move away from a technique like ultracentrifugation, because that's, it's serotype agnostic, and we come up with a lot of crazy serotypes, as does the rest of the field. So adapting techniques that are charge-based or basically non-centrifugation techniques to those to achieve similar product quality is a core challenge for us and the rest of the industry. But it's something that I really think about it as a really firm foundation that we've established to be able to innovate from, and that foundation is as Tim alluded to, probably the best producing AAV system in the industry. Of course, we could have a very fun and arcane conversation about measurement techniques and the justification for claims like that. But we have a system that is enough to enable CHF, which is saying something. It's certainly enough to be a credible CDMO player that's established itself quite well over the last couple of years. And it's been a platform that's been validated through a number of large pharma out-license and acquisitions now. So I think, without sounding too proud of it, we're pretty proud of it, and proud of what we can innovate from here moving forward as well.
Mm-hmm. Got it. Now that makes sense. And so in terms of scalability, you mentioned the commercial scale. Do you envision a need to scale additionally beyond the level that your commercial facility will be at, or do you think that's going to be enough and you just have more run capacity?
I think our scalability right now, we're not thinking too far beyond the limitations of single-use bioreactors, to be honest. We've been able in our clinical facility to pool multiple volumes of upstream quantities into larger batches. So we could release a 1,500 liter batch right now in our clinical facility, and the platform itself has gone to 2,000 liters, is operating at 2,000 liters in other contexts. So that kind of combination of rapid cycle times that we can enable with the platform, a pooling strategy if necessary, and then scalability to really the functional limits of single use as they currently stand. That's kind of the field of play as we see it. And if we need a 6,000 liter batch, we know we can do that. So far, it's an interesting dynamic where we're starting to see more of a call for smaller batches at a higher number. So we've had a number of interactions in a regulatory and in a customer context that have asked for, instead of a larger, 500 or 1,000 liter batch, is asking us to release a number of 250s to increase the N of batches going into late clinical. It's a bit of a weird dynamic to see that. It undercuts some of the cost of goods and efficiencies to reach the clinic quickly in some ways, but I understand why those customers are doing it. So I'm curious how that'll shake out as really commercialization becomes more commonplace.
Got it. No, that's interesting. And so maybe jumping to analytics, as you mentioned, that's an area where we've seen some conversation during this event as well with regards to incorporation of newer approaches, including droplet digital PCR, next-gen sequencing. Can you guys talk a little bit about where AskBio is with advancing different analytics approaches and where you envision moving forward in that?
Yeah, absolutely. So certainly ddPCR, as you already mentioned, is kind of a foundational technology that we're continuing to build on and really transitioning our programs away from qPCR to ddPCR, which we found is really probably the only way that we can reliably meet the sort of regulatory expectations that are starting to sort of come into focus now around the precision of our titer methodologies. So again, that's a platform that now we've sort of fully adopted, both within AskBio and at Virogen, and having a lot of success, I'll say, in terms of being able to establish as much as possible sort of a universal methodology for titering based on amplifying from the ITRs, which with qPCR is definitely problematic in our experience, kind of given the issues with incorporating the plasmid as the standard and so forth. So in ddPCR, we're finding that we get really reliable quantification of the transgene via an ITR ddPCR method. We'll certainly, sort of verify that, through some transgene specific, PCR runs as well. But we kind of really see a future here where at Virogen, from the CDMO point of view, we can have, a mostly universal method for titer determination, which meets the regulatory expectation for precision in terms of percent RSDs and so forth, and obviously becomes a lot easier to manage in terms of the whole method qualification validation life cycle, to sort of be able to leverage the platform. Again, we see it as now kind of becoming part of the platform along with the cell line and some of the other recovery process technologies and so forth. And the other key focus for AskBio, and again, by extension, Virogen, has been to really kind of try and bring these methods for characterization and QC into, kind of not even the 21st century, really the 20th century, in terms of, utilizing HPLC-based and CE-based methods for purity, getting real quantitative, purity methods where, we can actually start to do some robust sort of characterization and set specifications for purity that are meaningful, that are, quantitative and getting away from report results type of acceptance criteria, even for early clinical, as much as possible. Then certainly we'll need to do it as we move into pivotal and in commercial. So these are methods that we're developing largely at AskBio, certainly in collaboration with our colleagues at Virogen, and then transferring these methods, again, becoming part of Virogen's platform, to be able to sort of deploy these types of methods, not only to support our own programs, but to support third-party programs. And then finally, it goes beyond just kind of the QC, from our perspective. Again, we're really working to kind of implement that suite of extended characterization methods, things that I think, at least in my view, have not been commonly used, as part of regulatory filings and as part of that characterization package for viral vectors, doing peptide mapping and mass spectrometry to, assess sequence coverage and look for post-translational modifications, within the capsid and so forth. Again, you talked about next-gen sequencing, really getting to the point, as I kind of mentioned before, that we can consider these to be well-characterized biologics. I spent most of my career in the therapeutic protein space, and it was a little bit of a shock, honestly, to kind of start to understand how we were releasing product of vector and kind of the limitations of the analytical technologies. They really don't have to be there, right? I mean, we can do these things. It's just a matter of making that a focus, and then that's certainly one of our key focuses today.
Right, that makes sense. So jumping back to the upstream raw material setting, also mentioned doggy bone DNA, and so maybe you can talk a little bit about the advance of that and any other kind of attributes of raw materials and how that is shifting that could impact manufacturing success.
Yeah, absolutely. Thanks, Geulah. So I guess everyone knows, kind of the role that plasmid DNA has played, and still plays, right, in AAV manufacturing when you're in an HEK triple transfection mode. It's obviously a huge part of the cost of goods. It's also a huge part of the timeline, to actually get into the clinic, to get that material produced, for appropriate GMP quality, et cetera. And then there's, some inherent just risk factors with plasmid DNA. I mean, obviously these are bacterially derived, go through fermentation. That's part of the timeline challenge, right? It's costly and time consuming, to produce plasmid through E. coli fermentation. And then you wind up with a product that most of it is irrelevant and represents an impurity. And not only that, but a bacterial-derived impurity with origins of replication and antibiotic resistance genes and things that really don't have much of a place, in a viral vector product. So doggy bone technology, is a means to address all of those issues, with a synthetically produced, covalently closed, double-stranded linear DNA fragment that basically gives you the business end, right, of the plasmid and none of that other stuff that we don't need. So clear safety advantages as far as, again, eliminating all those bacterially derived sequences, eliminating the total DNA going into the AAV transfection as far as residuals, and also offering really significant advantages in terms of timelines, right? I mean, so we produce a batch of doggy bone in a matter of days, as opposed to kind of the weeks and months that it takes to produce and ultimately release plasmid DNA. So these are all the kind of values of it. This is technology that we identified as something that we would like to incorporate into our platform, right, as part of kind of the Pro-10 platform, and furthermore, again, kind of provide access to that technology, similar to what we've sought to do with Pro-10. So rather than just negotiate a license, to be able to leverage that technology for our own purposes, we sought to collaborate with Touchlight and establish Touchlight AAV as a CDMO business that again kind of gives access to this technology on a license and royalty-free basis, just a fee-for-service type of basis for clients across the industry. So again, it's definitely a key part of our manufacturing platform. AskBio is leveraging dbDNA, doggy bone DNA, in our clinical programs and preclinical programs, and we're certainly seeing really strong uptake and interest across the industry based on all these key considerations. Again, not the least of which is cost of goods, because data is proving out that doggy bone requires about 40% less DNA than plasmid. So there's a pretty strong driver from a cost of goods point of view, an even stronger driver, at least in my view, in terms of the timeline, being able to enable really rapid progression towards IND, without those complications and potential pitfalls of kind of getting in line, for plasmid at the players in that space.
Mm-hmm. Great. That's interesting. And so speaking of drivers of cost and other manufacturing innovations, are there other attributes of manufacturing that AskBio has incorporated or is interested in incorporating that can reduce cost drivers, in other areas?
Yeah, I think we're looking at this in a couple of different ways. I mean, there's technology to incorporate, kind of present state things like doggy bone DNA, kind of middle state, some of the downstream purification improvements, and then kind of long-term things like stable cell lines, those sort of true sea change advancements in how we make the product. You saw Tim alluded to, we try to have facilities that can adapt to those over time, so that no matter where the technology goes, we're fairly well situated to be able to continue to support the industry through Virogen and continue to supply our own programs. Part of it is also just facilities. We don't think by any means that we're done building capacity for Virogen. We've got a robust footprint in Spain for good reasons, and we've been really pleased with being able to work with the local, regional, and national authorities there to build high technology biotechnology businesses in that part of the world, and it's been awesome. But I think a U.S. expansion of that Virogen capacity is clearly indicated by the market, and kind of a sensible next step in I would say, insofar as construction is low tech, building more, it makes sense. So I think those sort of more of the same is also in the plans for us, for sure. I think the third part is just operational improvements. When you bring folks like Tim and other folks who've joined our organization in the last couple of years who manufacturing excellence is different than technology excellence. And I think the whole industry is going to benefit from the sort of manufacturing excellence practices. You heard Tim allude to, increases in throughput for us that didn't involve increases in capacity. Sometimes, our platform is really good. It's enough to supply diseases like CHF with a current footprint. It's just about turning a crank and running a really tight ship and incorporating manufacturing best practice on top of technology best practice. So I think those are really the three arms that we look at, right? Of true technological innovation, manufacturing operational excellence, and then just putting more square footage under roof so we can continue to build.
Mm-hmm. Great. And so looking ahead, the gene therapy space continues to evolve. Obviously, manufacturing is moving forward with it. What do you see as the next areas where we might see innovation in manufacturing, either along with or beyond the ones that you mentioned already? And also, what do you see as the remaining manufacturing challenges that you hope to see addressed?
Yeah, so again, I think we have alluded to, I think, many of these things. I think for most of us, I think we've kind of agree with what I think is probably a general perception that sort of, next and possibly final frontier, in terms of the upstream side would be stable cell lines, to be able to produce inducibly, controlled expression of the various, sort of elements to produce these viral particles. That's obviously an area of active development at AskBio, and again, we certainly do believe that's going to have a substantial impact on our industry in the years to come. Although I would say that we don't necessarily feel like this is an all or nothing type of dynamic, that we think that there's a place for certainly a strong sort of value-driven place for stable cell lines, but there's also a place in our industry long-term for maintaining triple transfection, especially with optimized reagents like doggy bone DNA, depending again on the indication and sort of whether you're talking about rare genetic diseases where you're dealing with incidence prevalence populations, versus again like a congestive heart failure type indication. So I think that's certainly a key element. I think again continuing to just again intensify the process as I alluded to, and increase overall productivity, become more environmentally sort of sound here, with the way that we produce these materials, as far as the detergents that we use, the plastics involved, the water usage, et cetera. These are the things that I think we're all going to be challenged with across the entire biologics and pharmaceutical industry to sort of do this in a more responsible manner, as far as the materials that we use and the waste streams that we generate. So I think that's going to be a key challenge for us in the coming years.
Got it. And so we've also heard a bit about discussion about regulatory trends with regards to manufacturing. Have you noticed any trends with regulators with regard to what they're looking for in CMC?
Yeah, absolutely. And I think we kind of touched on one already with the analytical discussion around the ddPCR. I think it was really just in the last year that we started, and again, most of our colleagues elsewhere in the industry have gotten the same feedback that the regulators are expecting precision and percent RSDs, less than 15% for the titer method. That frankly for most qPCR methods is not routinely achievable, right? So that's really I think the move towards ddPCR obviously was already happening, but I think that's really kind of pushed us even faster down that road. And then just more generally speaking, I think as more and more of these products are entering clinical development, I think the regulators obviously are aware of this potential dynamic, again, depending on the indication, for these products to move very rapidly from a phase 1, phase 2 study to a registrational type of study. And so I think, we're starting to see that the regulators are asking more questions earlier on, that even at the IND stage, we're dealing with requests and inquiries and questions that one might expect, a little bit later on in development, which I think makes sense, right? I mean, if we really are going to be able to jump, straight to pivotal studies based on a really exciting, phase 1, phase 2 readout, we have to be ready for that. So I think as an industry, we need to start to adapt and kind of be in a position where, we have a solid CMC kind of plan, even, at phase 1, phase 2, this idea that, we're going to start, with a very rudimentary adherent type of process. We're just never going to be able to then get there, at the time pace that we'd like to if we're not really, have an integrated CMC approach. And to that end, one of the advantages I think we have, with ViralGen and with our platform is, ViralGen has a master file that I think is a real kind of risk mitigation and just timeline advantage for its clients, to be able to leverage and reference that master file to incorporate some significant elements of the CMC package. It's obviously not everything, but I think it really creates a significant de-risking and timeline advantage. And that is too, like everything, is something that just continues to get optimized over time, right? As we continue to get feedback, whether that's through inspections, regulatory inspections, client audits, or just the general feedback we're getting in Type C meetings and whatnot, to sort of leverage into that master file.
Fantastic. That's great. So I see we're just about at the end of the session. So I wanted to thank you both for the fantastic discussion and for the participation.
Thank you.
Thank you very much, Geulah.